at it.
Oh, you got the link?
Yeah.
Lovely. Just check.
But that's only for CNRS. Wait.
No. We need it for everyone.
That's weird. It changed on the website as well. Like on the Nature website, they put the link for so weird.
I know. It's because
in the DOI, it's so weird. Hello.
Hello.
Good morning. Sunshine.
Hi, Sarah.
Boom. There is the link.
That is looking good now.
And I haven't downloaded the paper.
Great. Are we ready?
Yeah.
Okay. He disappeared. Anyways, well, welcome to Neuroccino on this Monday morning. It's the 30th of May today. The way Neuroccino works is everyone is welcome to join us, present a paper they got excited about, and just tell us why you got excited about that particular paper or book chapter or YouTube video or any piece of science that got you hyped. Today, Michelle is going to present a paper to us. Don't actually know which one, but Michelle, if you're ready, if you want to share your screen.
Sharing my screen. You know which one, because you're the one who sent it to me 2 weeks ago.
Great. Oh, that one. Yes.
Yes, that one.
Hang on. Can you go
you
full
sent to me.
screen, please?
Yes. Is it better? Is it better? That's better.
Whoa. That's good. Thanks.
All right, so this morning we're going to talk about the emergent reliability in sensory cortical coding and interarea communication, which is a paper that have been published 2 weeks ago. So it's very recent. So why did I get excited? I remember Crosscorrelation saying, if you were to come on our planet and do some research on the artifact we left on the planet, and you find a book, there will be different approach to try to understand what this book means. And you will have people doing science that are reductionists will take the book, shred it into pieces, identify all the atomic composition of this book to have the pure description of what is the book and report it into an article explaining that they finally, shed light onto what a book is, but will never, ever understand what this book was about. And in contrast with that, you have people that are trying to look more like at the interaction between different elements that in this kind of metaphor, we'll open the book and try to look at the interaction and the repetition between the letters that will allow us to extract words and understand what the meaning of the book is without technically knowing what its composition is. So how does this extend to the brain? When people have been doing cellular imaging and also the field of cellular imaging, due really to a technical limitation, it's been really focused on the recording of single cells in the brain. So you have not that many papers that can capture enough cells in the brain to start looking at interactions. And in the present case, the authors managed to measure many cells of the occipital cortex, 21,000 neurons, at the same time doing a go-no-go task that mice have been doing. And that does represent the entire visual cortex. And the go-no-go is, they call it a visual task as neuropsychologists. call it a decision task, but in the present case of mice, I guess this is a visual task. And essentially what they've been doing, the screen is stuck. All right, so they take those little mice, show them these tasks. Sometimes they have horizontal grading, sometimes it's a vertical grading. When it's horizontal grading, it is actually a go signal. So they either hit the target and they're gonna get positive feedback, which is like water, or I don't remember what kind of food they give to them. If they miss it, they have nothing. And the no-go signal is this vertical grating that you have here. If they actually touch the screen, when they do that, that's a false alarm, and they will have a little puff on their face as a negative feedback. It's really annoying. I don't know if you get puffed in your face in your life. I don't like it at all. And if they don't do anything, it's a correct rejection. They don't get anything, and that's absolutely fine. How was it possible to record all these cells at the same time? Well, this is a technological breakthrough. They've been using. Sorry.
Sorry to interrupt you. So the response was, what was the response of the mice?
I think they're leaking the screen or something like that. I don't remember.
Oh, okay.
So that was only possible thanks to a great technological breakthrough, which is this imaging of calcium activity through thousands of layers of pyramid neuron using a custom microscope. They built a machine and they played around and they measured all these neurons, as that you can see here in the visual cortex, and they can have it in real time. And there is a video and supplementary materials that I downloaded for you. Just gotta find it. Up. This was video right here. Up. Can you see the video? So you can see neuron firing over there in real time during the task. And I don't know how good your eyes are, but to me, if I was to try to guess what was the code that was behind that, coding for what mice have been seeing, I wouldn't be able to do it. It looks totally random. And so that's a question that I try to assess, is how to use all this information to decode what the mice have been seeing. And so the first thing that they try to do is to measure single cells. This is very slow. Okay. And look at how they are preferentially associated with a different kind of the two stimuli. So the first thing that they find is like many cells of the visual cortex, as we know, preferentially responded to one of the two stimuli, either the go or the no-go. And that's great, and that's totally localization of function and single cells, except that they have many at the same time, and they can see that there are a lot of cells that are preferring one stimulus over the other. But the sticky part, because they've been recording those mice for an awful long time and several times, they have a 280 minutes duration of videos. They notice that, like the cell properties, they change between sessions. Sometimes, those cells prefer like one stimulus, and sometimes they prefer another one, even though it is exactly the same cell. And you can like, well, where is my functional specialization there if between two sessions now my cells are not anymore liking the same stuff? And this was way higher the signal noise ratio. So it's not like a problem of like the method is not so sensitive, and we sometimes things this difference because it's a noise effect. It is a true intracell variabilities that do change the property across time, and that was a real bummer for this method that they use, that is a decoder that helps them to decode what actually the mice have been seeing. Because if the cell change their properties, you can't decode anymore what the animal was seeing just because the cell are not doing the same thing anymore. And so that's where they thought it'll be probably cool if we try to, instead of looking at all these cells to decode, try to capture the interaction and try to see if the interaction between those cells actually predict better the outcome, what the mice have been seeing, rather than the individual cells. In some, they've been doing functional connectivity, but at the level of single neurons with calcium imaging. That's what they've been doing if we would like to compare it to what we're doing as neuroimagers. And so, you know, the article is quite complex and I didn't have so much time to work on it, but I found this really interesting because it took the decoder and looked at the correlation between the decoder of different cells. And you can already identify here this kind of matrices that we find usually when we're doing our correlation across time, particularly when we're doing EEG or MEG. And you can see that you have territories where the correlations between cells is extremely strong and correspond to the lag of time before the appearance of the stimulus. That's when the stimulus appears, the delay, and then for the response. And they've been using this to try to decode the activity of the mice. So there was a mean similarity between all pairs of instantaneous decoder assessed by a correlation coefficient between pairs of decoder weight and all the cell of each mouse, given the decoder constancy across stimulus presentation. That was what I just showed to you. And what they found, although I'm still looking for the chart, is like accounting for correlated fluctuation among task-related cells facilitates stable representation of stimulus type, which is a demonstration that looking at the correlation between the cells in the brain is better to represent what actually, or decode what actually people have been seeing, what mice have been seeing, rather than looking at the activity of single cell, which means that the emergent property of the interaction is actually more meaningful in terms of decoding what people see. So it reversely would suggest that it is more meaningful in terms of how it is coding what those mice have been seeing. So I just highlighted this because I find it interesting. We often do functional connectivity, assuming that areas are directly connected. It is not the case, and they know it as well. They've been writing it. Activity cofluctuation, which is what functional connectivity is, of cell ensemble are thought to reflect shared connectivity, such as common input or direct interconnections. But you know, it's one or, you know, if that's a common input, that doesn't mean a direct connection. Okay. Do, and those, that is the chart, I think, that demonstrates the best, the quality of the prediction based on the cross correlation between the encoders. And you can see here that the coding of the stimulus is actually extremely high over here when you look at those cross correlation between the different neurons, demonstrating the integration, the relation with the integration, and the coding of the visual information. So in sum, this paper have been demonstrating that by tracking neurons across all visual cortical areas, they reveal that information processing mechanisms are likely to underlie reliable sensory performance through the correlated fluctuation that facilitates the long-term reliability of neural population activity decoders. That's it. If you have any questions.
Great, thank you, Michel. Any questions? Just put your hand up and we can ask Michel to specify, clarify, whatever. Victor, you have your hand up.
Yeah, that's a very interesting question. I have two questions. One is very methodological, and I don't know if you have the answer, but I'll start with the first, which is what exactly are decoders? I didn't get that precisely.
So my understanding of the decoders is a way to take the activities that they record and use it to predict what the mice are actually seeing, whether this is go or no go.
So this is some kind of machine learning thing that tries to interpret the signal from cells and to say, okay, this is related this way to the input time series or something like that.
So I look at the supplementary material and there is some like equations onto like the prediction, but I didn't check whether this was advanced machine learning approach or it's a simple linear regression that they
doesn't need to be advanced. I mean, machine learning is maybe sometimes simple, but yeah.
Well, I guess if they were to use a advanced machine learning, that will capture the interaction between the neurons and show a very good prediction, except that they wouldn't know that this is an interaction between the neurons that is better predictive than single neurons. So there might be a simpler algorithm. I didn't look into that. I tried really to get the gist of. But the supplementary material is like 4 or 5 pages, so you can easily go through and find your information.
Yes. And my second question, but we'll see if you know, it's like 280 minutes is very long. They did that for each and every mouse, or it's like in total?
It's a good question. So that I think that's per mice because the video is 280 and it's split into different sessions.
Okay, because one thing that I found astonishing is I worked with this kind of, well, not exactly this one, but the same kind of fluorescence activated by usually transient calcium signal. And there is a big photo bleaching effect from repeated measure. And if they managed to do that for a long time, like how do they do it?
They did mention it in the text. Because I never used this method, I was like, oh, it's all right. But they did control for the photo bleaching. It is somewhere in the text. I didn't highlight it. I'm sorry, but they talk about it. I remember seeing it. I just didn't see the relevance of it because I never used that method. But now that you're asking for it, it's in there. It's in there.
Okay. All right. Thank you.
Ma also has a question.
Yeah.
Yeah, thank you. It was very interesting. So my question was because they find that there is no, it's difficult to predict based on the individual activity of the cells. So I was wondering if maybe it reflects better the learning and practice effects. This individual variability, it's reflecting the change in due to the learning and to the practice.
It's a very good question. I don't know. I'm like useless every question. I have no idea. One thing is sure is the process of learning is not going to be done by changing your cellular type. it's going to be done by changing your connectivity or the weight of your connectivity. So if we looking at learning, then we actually getting back onto like emergent property of the interaction between different cells, which is the main message of the paper. Now, yeah, I guess, you know, you're exposed to the same task a lot of time and your brain adapts and does it better. Although the mice have been trained a lot before starting doing the task, because they have to do a go no go, so they have to restrain themselves until the task start in order, you know, before they try to get the positive feedback, which is just a tube from which they can get, you know, like a juice or whatever they give as a positive feedback to those mice. But I mean, you know, the frontal lobe of a mouse is not that big, and pretty sure for a long time they were licking the tube right away during the entire session. So training them is actually a big challenge. And they were trained to the task as well, because they're able to do the go no go, which, you know, I don't know how they managed to train the mice to do that, because to try to, you know, ask a marmoset to do that, you're going to spend a long time. And a marmoset is like way more complex animal than a mouse. So it is actually from the technical perspective, a beautiful paper, and from the message that they give, quite an advanced demonstration that is encouraging for people that are looking at connection as a fundamental basis of the functioning of the brain. And to come back to your question in terms of training, because they've been trained so much, the assumption is, you know, you reach a plateau where doing the experiment more shouldn't change your neuron, but there is no way to control it perfectly. But that's this information that the same neuron that was very sensitive to one kind of stimulus, and that suddenly just changed, has never, to my knowledge, never been demonstrated at that level of resolution when we can actually record single neurons. It was suggested before through brain stimulation, or people like had a single neuron and then it stopped to being responsive, but they thought the neuron died, or, you know, but yeah, that's an interesting information, I think.
Yeah, thank you.
That actually links to the question that I had for you. So this paper was published in Nature.
Mm-hmm.
And I was wondering if you could summarize for us what the novelty and the excitement and the real impact of this is.
All right, the novelty is that the functioning of the brain is not in single cell. The functioning of the brain is in the interaction, the emergent properties between cells. That's the big, oh wow, we understood something that we didn't understand before. And then there is all the cool part with the advanced techniques, these videos that is showing the activity of neuron during the task. That's pretty advanced. I have to say that for a non-expert reading the paper, the introduction was clear, the discussion was clear, but when it gets into the result and the method, it was rough. It was a rough ride. But you know, that's what we need for. It's like to learn, and I find it very interesting.
Leah also has a question for you.
Yes. I was really just fascinated about the resolution that these measures should have in time and space. I imagine that they are super higher respect what we can measure, for example, in an MRI or fMRI. And I was thinking, how can we merge these two different informations and from these two different scales?
Well, you know, using the same species would be already a good beginning, because you cannot really match a mouse brain onto a human brain. Sure. But let's say we stay within the same species. Choice of heart, we'll go for the squirrel monkey.
Okay.
We'll be able to measure that. So you do well, you can do an alignment between the method, but it takes time. So people really like the automatic approach, but technically, you know, this will require manual alignment between the different dimensions. And then if you want to capture the interactions, it'll be really looking at huge matrices of information. So there is two steps for this. Step number one, you need to be able to register perfectly your different dimensions, whether this is calcium imaging or MRI measures, or you know, any kind of, or postmortem, you know, histological staining. And then the next step is to be able to extract from this information matrices that are matched in that space in order to capture the interaction between the different dimensions. I guess the third step would be to bring it back again to the brain to have a cool picture, but this is just fancy imaging. But the registration between the dimension is a sticky part. I've been doing it a bit, and it's really hard. There is no better way than doing it manually, just because the information are so different, but your eyes can actually detect the match between different anatomical landmarks, and that's how you do it.
Okay, so Frédéric, I was thinking that maybe we can like analyze these matrices of interactions in the two different resolutions and then try to learn something from these.
You can actually try to run correlations. That's, you know, will be it'd be exactly like doing simultaneous fMRI, you, me, and try to see whether there's correlation between our brain areas and not aligned together. But we can assume that because there is correlation, they're supposed to be aligned. But this is an assumption. I personally like anatomy, so I think we should start by registering carefully landmark together. But you know, you can have a computational approach and try to look at the mutual information and assume that because there is a strong shared information, this is a way to align information together.
Okay, thank you.
Valentina also has a question for you.
Do you think this pattern of activation could reflect the activation also in other type of cortices and also in other type of tasks like higher order cognition, rather than just sensory tasks?
This is a very good question. So often we assume that because we decoded for one function in one brain area, it is the same functioning for the rest of the brain and every other species. No, we can't assume that. It is just one example. I think the visual cortex is a nice touch because this is probably the cortex have been the furthest explored in terms of localization, hierarchy, area A speaking to area B, speaking to area C, giving you, you know, the decoding of this word, and showing really that interaction between brain region is more meaningful, that the activation of single area over there is a nice proof of concept in a domain where people were so much anchored into extreme localizationism. That would have been the frontal lobe and decision making. People will be, but we all know that it requires more than. But in the occipital cortex, it is really like compartmented. So I guess it is the right place to make the point, but inferring that this is the same for every species, every function in the entire brain would be clearly an overstatement.
Thanks.
That is all the questions I can see, and that's all the time we have this morning. So thank you very much for this great presentation, Michelle. The slot for next week is still open, so if you come across an exciting piece of science, let me know, and it's all yours. Everyone is welcome to present, as I mentioned earlier. And that leaves me to wish you an exciting week in science, and see you